And so our first panel this afternoon is led by Bob Holt, and I don't know how many times Bob has been to NCEAS things, so how many people in the audience have been in working groups with Bob? Yeah, okay, pretty large fraction. So he probably really doesn't need any introduction. But he has been on a large number of working groups here, and is going to lead this panel today on ecological synthesis for human well-being, environmental dimensions of disease dynamics.
Right. Well, it's great being here and seeing so many old friends and making some new ones along the way. A word about the panel composition. And when I realized Andy wasn't going to be here and I wasn't sure Peter was going to step in, I said, oh, maybe I should, this is California, New Age, I could try to channel Andy Dobson, but that would be dangerous probably. So I asked him to send me some notes when I found out he was feeling well. So I'm going to blend just a few thoughts he had with some thoughts of my own. So as we go through, I think we'll each have a short statement regarding the role of NCEAS in the area of environmental dimensions of disease dynamics, and then sketch some key directions for future inquiry and understanding. It's my take on the discipline, and that of Andy and I think the other panelists as well, that NCEAS has played an absolutely catalytic role in bridging epidemiology and ecology. The time of the founding of NCEAS, 1995, was very propitious in terms of the development of these disciplines. On the one hand, there was growing recognition within epidemiology and the public health world that you needed to incorporate a richer ecological perspective, particularly when you're dealing with zoonotic diseases that could spill over from other organisms in the environment. And on the ecological side, there was also growing recognition that we had been collectively ignoring many hidden players in ecological systems, parasites, pathogens, and for that matter, other microbes, mycorrhizae and the like, things that are kind of hard to see and study when you go out with your quad bats, your binoculars, and so forth, but could be really very important. So the first working groups of NCEAS that I could find, I found about 22 in the records that seem to be related to infectious disease biology one way or another. But Cornell had one that was relevant, but that was sort of a smallish working group as I recall. You had two, he corrects me. And then Rick Osfeld, who's not at this meeting, played a really important role by setting the first large working group that was sort of multifaceted, specifically on interspecific interactions and biodiversity in the context of the ecology of infectious disease. And one thing that Les pointed out to me in a discussion a little earlier, so it started with this sort of large working group, but one thing that evolved out of that was that it wasn't just a one-off activity. There were a whole series of working groups over time, and in fact, one thing that happened during those early meetings was sort of a plan for follow-up, multi-year activities. So there ended up being working groups on infectious disease and conservation biology, on spatial dynamics in infectious disease. Later on, topics like seasonality, the ecology of ocean systems that Drew will speak of, and so forth. And I think that's one reason that NCEAS had such a catalytic role, was that it had a series, a sort of a continuity over time of interwoven groups of people who then went out and carried on in their own way, stimulating the NSF EID program and various other initiatives that go well beyond anything NCEAS itself could do. One thing Andy did point out to me that I think is really very important, and one reason I think that these NCEAS working groups have been such a success in terms of publications and career development, is that even though involved silverbacks like Les and myself, and Andy,
thanks.
I'm not mentioning you, pointedly not mentioning you, but that we didn't dominate the discussions. There were postdocs, there were graduate students, there were younger faculty who were really very active, and if you look at the papers that were produced by the group, usually it was one of them that was first author, and often the bulk of the authorship was made up of sort of younger people. And I think that's one of the really valuable things that NCEAS has done. It's not just sort of stimulate the science, it's actually got this tremendous role in training people And that was instantiated in our working groups. Now you think about, you know, just in terms of ecological contributions that have come from this, you know, if you think about classical epidemiology, we all know, any informed citizen knows that diseases have had a huge role in human history. You know, Alexander the Great's invasion of Asia stopped because of a malarial fever that killed him. You know, Cortez conquered Mexico because smallpox was sweeping through Teotihuacan, laying waste an army that was much larger than the band of 200 Spaniards that Cortez has. The Europeans conquered tropical Africa, but they didn't colonize the lowlands because they were kept out by infectious diseases. So there are major swaths of our own history and our own distribution on the planet that have been driven by interaction with infectious diseases. Why shouldn't the same be true in the rest of the world, rest of the biosphere as well? And one of the things that's emerged from working groups led by Andy, Rick, Les, and others is that, for instance, you know, parasites make up, after all, between 60 and 90% of all species on Earth, very large slice of biodiversity. But if you look at food webs, it turns out that you're missing about half the interactions that are going on. The feedbacks, the direct density-dependent feedbacks of a species on itself, and various kinds of indirect interactions due to shared pathogens, due to pathogens facilitating trophic flows of energy and materials by making prey easier to catch, and so forth. And sometimes, even in terms of just production and total biomass, that the Carpentaria Marsh, there's a substantial fraction of the biomass there is actually hidden in the form of parasites hanging out in snails. That has really, I think, opened the eyes of lots of important research questions where we revisit some of the classic themes in ecology with fresh lenses and ask fresh questions about them. So the whole stability complexity debate has to do with how networks of interactions play out in response to perturbations. But if you're ignoring a lot of those links in that network, you may be totally misled as to what's going on. If you think about what maintains biodiversity, I've been talking to Peter Chesson about this. He's convinced it's actually mainly a food web phenomenon, where there's things like habitat partitioning and so forth, but greatly modulated by pathogens and predators along the lines of the classic Jansen-Connell effect. So I think we're going to be revisiting a lot of crucial classical themes in ecology, but let me end my comments just by saying a little bit about where I think we need to go. So here are two directions that I think are inevitable and exciting, and possibly NCEAS 2.0 can be involved in some of this. One has to do with the issue of scaling down as well as up and understanding host pathogen interactions. If you take a typical bacterium, it's about a micron, i.e. 10 to the minus 6 meters in size. Scale that bacterium up to the size of you, the human host, 1 to 2 meters, okay? How big is that host if you scale the host in like manner? It's about 4 or 5 times the size of California. It's an enormous area. And so if you think about the host as being this sort of landscape within which pathogen dynamics play out, all the processes that we're familiar with in standard ecology, things like source sink dynamics, habitat specialization, resource gradients, and so forth, kick in and understanding what's happening within an individual host in terms of its infection dynamics. And I think there's a huge arena there. And in fact, you know, were I to sort of project what an NCEAS 2.0 might have, not just sort of human health issues and animal and plant diseases, but microbial ecology in general might be a much larger player over the next decade than I think it had been over the last 16 years here. And then the second thing that I think some of the other panelists will speak to is, of course, evolution. You know, pathogens and parasites have large population sizes, short generation lengths, a lot of genetic variability. They can evolve in real time within an individual host and certainly among host populations. And when I think about ecology, where I think we're going as a discipline over the next 20 years, we're going to see an increasing intertwining and fusing of ecology and evolutionary biology at all scales of analysis. and nowhere more than in the study of infectious disease. And with that, we're going to walk through all of us, and then at the end, open it up to questions and discussion. Drew.
Thanks, Bob. Thanks for your comments as well. So it's really a privilege to look out across this group and see so many old friends and to be here. I really appreciate it. And it was fun to think back over my time at NCEAS in spending my sabbatical here in 2000, and then subsequent working groups. And I don't think it's too dramatic to say that my time at NCEAS turned me from a quantitative field marine ecologist into an epidemiologist. I mean, it was unbelievable to be here during the years between 2000 and 2006 with this just massive profusion of disease ecology working groups, from working groups focused on spatial ecology of disease processes, diseases and conservation. And I look around and I see some people, nodding that were involved in those. And so it was phenomenal for somebody like me who was just starting to study disease ecology because of our changing ocean and what was happening, and the increases in these levels of disease. So I wanted to think about what I could say that would reflect on the past and inform the future a little bit as we were tasked to do. And I thought what I would choose for an organization is I would go through 3 papers that came out of working groups that I chose as being sort of the most important ones, or the ones that have a history that carry through to today, and then also reflect on where we are now with those. And fortunately, one of them is in that list. I was really worried. Oh God, the list. What if it's not in there? So I'm sure everybody has a paper in that list. So in 2002, we published a science paper reviewing how climate change impacts were already increasing with warming conditions. And this paper was kind of important to me because I had an overly simplified vision of how this was going to go, because I've been working on coral reef systems. And we were struggling with the starkly shocking events of coral bleaching and subsequently, kicking off these rounds of disease outbreaks. And so, to me, a warmer world was clearly going to be a sicker world. And I would like to give Andy Dobson credit for that. He's the one that thought that up. But of course, in making the extensions to agricultural and wildlife conditions, it was far more complicated than just a simple warming event. And so I think that paper raised a lot of questions and a lot of interest in the complexity of ways that climate change was affecting disease outbreaks. Now for me, as I said, I already knew warming was going to be the issue of our times because we'd already been dealing with it for, at that time, about 5 years on coral reefs. But I was nowhere near prepared for the pace at which the future was going to come to us. And I think all of us in this room are probably a little shell-shocked, at least that think about these things, for how rapid and extreme some of the climate change impacts, both on health, ecology, sea level rise. I mean, it's just overwhelming the juggernaut that's bearing down upon us. And at the time, nor did we understand that ocean acidification would create the second arm of a pincer with warming that would spell disaster, at least if things continue, for an entire marine ecosystem. So following that paper, we developed forecasting tools using a lot of the skills in quantitative biology and modeling that we learned in the working group for coral disease, and that actually did lead to a national program today in Australia for forecasting, and then subsequently what you do about disease outbreaks that are temperature triggered. And then to bring us up to the current, last week we just heard about our new NSF grant to launch a research collaboration network on marine disease climate impacts for the social sciences integration. So that's kind of my story of how that one paper kind of unfolded through time, and really that very much was kicked off in actually Andy's diseases and conservation working group. So in 2006, Kevin Lafferty and my student Jessica Ward in our working group published The Baseline of Marine Disease, again a literature analysis detecting increasing reports of disease outbreaks in the ocean. The working group correctly detected this rising tide and the rapidity of disease transmission in the sea, but not really what to do about it. And I think we're still left to this day saying, you know, how do we cope with both the detection and the management problem that these poorly understood ecological processes in the ocean present us with. So for today, on that one, I like to think there might be an opportunity with a new effort that Ben Halpern and Karen McLeod have been developing called the Ocean Health Index. The OHI, as it's called, is currently conceptualized as a series of 10 human uses in the ocean to develop the index, but none of them includes a component of organismal health, even though it's called the Organismal Health Index. And so it's very nicely targeted on positive human uses such as fisheries yield, the positive attributes of a healthy ocean, or ecotourism value. But I think that, you know, this is a challenge to develop the organismal health layer to be able to populate something like a management policy tool like the OHI is intended to be. So the last memorable paper, and this one actually didn't come out of a, well, these were memorable to me. I hope they were memorable to some of you. This one didn't actually come out of a working group, but I like to refer to it because it came out of a whole series of people that met in the working groups, that worked closely together, remained as collaborators, and then when the time was right and you know, an idea emerged, came together and spontaneously wrote this paper. And this is the one Felicia Keesing's the senior author on, and the tagline on that, I guess it was from Time, was that biodiversity is good for your health. And so this was a paper that developed some of the ideas, actually, that, you know, Bob was also involved in this, about how the complexity of the host community can affect the rate at which diseases are transmitted, not only among wildlife, but also even, in fact, to humans. And so there are social issues associated with understanding the ecology of transmission of disease. Now, less said, don't open this issue. There are people in the audience who really are a little critical of that paper. It's considered to be a bit controversial. I think in part it's controversial because our understanding of the transmission biology of diseases is still really in its infancy. And that's even on land. I mean, we're challenged to even get it right on land. And the really I don't know, discouraging aspect of that paper for me was that we have no idea in the ocean how the biodiversity of hosts affects the transmission rate of diseases. We don't even know who most of the hosts are in the ocean. And so if we're going to move forward to solve some of our really important pressing issues like food in the future and rely on the oceans as one way out and the potential for aquaculture in the oceans to help us feed the future, we're going to have to be able to understand these host pathogen interactions so we don't have these huge billion-dollar problems like salmon viruses wiping out Chile's salmon farming efforts. And so I guess with that, I just want to, I'm going to wrap that up because I'm getting science from the back. Those are my three themes. Thanks.
Well, I took as my charge to talk a little bit about the relationship between developments in disease ecology, NCEAS's role in that, and collaborations with and extensions into the world of public health, because that's where I work in now. And it's important to recognize that there were precursors to NCEAS's activity. Disease ecology wasn't born at NCEAS, and in recognizing that, one can try to figure out what NCEAS's role really was in this. The SBI document, as Jane pointed out last night, tried to articulate two dimensions. One dimension for scientific foundations and another dimension associated with national needs. And I was part of that as well as Pete Peterson was part of that. We were talking about it over lunch. And it was very early on in the SBI that we recognized that disease was an area in which ecology would be able to contribute significantly in both domains, because pathogens had been ignored in the structure of ecosystems, and also people that worked in infectious diseases primarily were not using ecological methods or ways of thinking. So we identified disease as a major research focus in the SBI, and that developed fundamentally into a workshop that was sponsored by the Board on Environmental Studies and Toxicology, jointly with the Institute of Medicine, which I chaired with Rich Lenski. And NSF had participants there, but the Deputy Director of the National Institutes of General Medicine and the Deputy Director for the National Institutes of Allergy and Infectious Disease were both there. And at breakfast, one turned to the other and said, "I thought you funded this kind of research." And they said, "No, I thought you funded this kind of research." So at that point, they decided to establish the first program announcement for the evolution of infectious diseases, which included ecology at NIH, which was a joint venture between NIH and NIAID. But NCEAS was really critical in coalescing a community of ecologists who were interested in this broad approach and applying it both to scientific foundations and the area of public health. Bob mentioned the first working group, which was kind of a coordinating committee that got together here at NCEAS that Rick Osfeld pulled together, which involved a lot of different people from a lot of different communities. And that was one of the things that I wanted to highlight about NCEAS. Other people have kind of mentioned it, but it really became important in this field was that it's not just the synthesis of data or ideas, but it's the synthesis of different discourse communities, of bringing entirely different intellectual communities together that never ever interacted. So Rick brought people in from CDC and from epidemiology and public health, as well as classically trained ecologists, to sit down in the room together and try to figure out what we had in common. And there were many projects that spun out of that. And that was the other main contribution of NCEAS, and that was also a deep commitment by Jim Richmond of saying, well, this is an area that we think might have payoff. This is something new, and it has outreach as well as foundational interests. Let's organize a set of working groups around this general topic. So it was a general risk-taking to commit deeply to try to develop a particular area that may have paid off. And I think Jim deserves a lot of credit for taking that risk. And we hope it paid off for him, but we benefited from that level of commitment. So disease ecology has, just for people who don't know, NCEAS helped stimulate a whole community. When I organized the Symposium on Disease Ecology at the Ecological Society many years ago, and it was at a time when Andy Dobson couldn't even get his abstracts accepted to give a talk at ESA because the subject matter was viewed outside the domain of the ecological sciences. Now we go to the Ecological Society meetings, and it seems like almost every other session has got some aspect of disease in it. It's really kind of getting boring. But it's been influential. There have been the developments and intrusions into public health. There's now not only the joint effort with NIGMS, the General Medicine, and AID. There's a collaborative activity between the Department of Homeland Security, Fogarty International, and NIH called RAPID, which is Research and Policy on Infectious Disease Dynamics. That's an activity that's spearheaded by Brian Grenville and Alice McKenzie. There's a joint NSF-NIH program primarily organized by NSF on the ecology of infectious diseases. NIH also has a separate program called MIDAS, which is on mathematical modeling of infectious diseases. And the deputy director of the Institute of General Medicine said to me a couple days ago, well, not a couple days ago, it was a while ago, that to quote her exactly, she said ecology is no longer a dirty word at NIH. And once you get over your anger that they thought it was a dirty word, once you get over that, you recognize how important that's been to get this kind of, because the biomedical sciences are the 900-pound gorilla in the scientific community, and to get that kind of level of acceptance is really, really important. And I wanted to finish by saying that I think that we're at the beginning. So that's the looking back part. We've been successful. How do we look forward? I think it's to take this general approach and recognize that we have a lot to offer in that community, in the biomedical community and in the public health community, and that we should not be shy about extending our approaches and our understanding to whole new novel areas. So I think that we could, in fact, start talking about the general ecology of global health as a domain for our concerns. We can start talking about the ecology of violence, the ecology of displaced populations, the ecology of safe water, the ecology of poverty and hunger in the world, all of which have very tacit and sometimes quite visible ecological dynamics which underpin them. So I think NCEAS has played a really significant role in creating those bridges that were never there before, very important ones. And I think that there are many, many more bridges that we can create in NCEAS too. So that's my story, and I'm sticking to it.
Thanks, Liz. So I'm not Andy Dobson, obviously. I don't even have a leather jacket, so wouldn't be able to channel in very well. So NCEAS certainly, there's no doubt, has contributed to a range of cross-disciplinary activities. I would suggest in disease. Bob has mentioned a number of those, and Les and Drew have talked about others. I was involved a number of years ago in an NCEAS working group on trying to understand the impacts of host-social and mating behavior on, I guess, the ecology and evolution of parasite communities and pathogen communities, and trying to develop better predictive understanding of how behavior might influence those communities. And that group was interesting to me because it involved a range of different people. It had human sociologists in it, veterinarians, population biologists, and even I think a taxonomist or two. So it was really quite a diverse group, and a really good example, I think, of the kinds of people that NCEAS could draw together to tackle interesting issues in ecology. Since then, I know there's been other working groups that have tried to bring together, I guess, to go further down that track, to talk about, to try to develop models for understanding how environmental heterogeneity influences host behavior, and then, for example, how that then maps onto patterns of disease prevalence and incidence in space and time, so that sort of, I guess, topic of behavior and its intersection with environment is one that's played a role in several different kinds of activities here. But of course, behavior is only one aspect of a broader consideration of how life history variation might influence interactions between hosts and pathogens, particularly in terms of thinking about, not just in terms of the basic sort of community and population dynamics of those organisms, but in terms of thinking about our ability to predict invasive pathogens, emerging pathogens, or what might be likely to be effective biocontrol agents in different situations. So there's a strong applied aspect to understanding those kinds of issues. And I guess something that interests me, probably coming from more of an evolutionary perspective, is that I think it's still remarkable that we still have relatively little empirical data on direct links between genetic variation at loci that are directly involved in these interactions and to spatial patterns of disease incidence and prevalence in wild systems. And that's, I guess, one of the sort of directions or possible directions I think that NCEAS could be heading toward, is starting to try to integrate the ecological, environmental, and genetic dimensions of trying to understand these systems. And so really connecting genetics, demography, and environmental factors. So I know earlier there was a lot of discussion about bioinformatics or ecoinformatics, but obviously there's also increasingly this incredibly powerful sets of high-throughput genomic tools that we've developed that are going to allow us to tackle a lot of issues as well. And I guess an interesting challenge to me would be how do we bring together ecoinformatic and ecogenomic or metagenomic kinds of technologies to link our understanding at different, I guess, spatial and temporal scales of organization in these systems. So Drew talked about climate change, and that's a major player, obviously, in what's happening. I guess I would suggest that there's some other major kinds of change that are going on all the time as well, and that these sorts of, I guess, global patterns of change that are occurring are particularly critical for understanding antagonistic biotic interactions such as pests, pathogens, and so on. So, for example, climate change, yes, that's absolutely a major element of change. Another really important thing to think about, and this is particularly true in terrestrial systems, are food security issues. So we know that in the next 30 to 50 years, for example, we have to double our food production. Global food production has to double in the next 30 to 50 years to feed the population. Well, that has all sorts of knock-on consequences in terms of changing patterns of land use, agricultural intensification, increasing deployment and acceptance, I suppose, of GM technologies, use of nutrients, etc. And those kinds of things are all factors that have the potential to tremendously impact on biotic interactions in these landscapes, particularly including pests and pathogens. And I think it's important to think that within that we might start we need to do a better job, I would argue, of working through how to apply evolutionary and ecological principles to managing those interactions in these ever increasingly fragmented and managed sort of landscapes. So there are a few examples where that happens, but not that many yet. So the deployment of GM technologies in cotton, for example, BT technologies. There's a deliberate attempt to bring evolutionary principles into managing those kinds of interactions, but it certainly hasn't happened in a lot of other cases. Also, I guess connectivity. There's a recent analysis, I read an article about basically showing that at this point in time, less than 10% of the world can still be considered to be remote. So increasingly, we live in an incredibly connected planet, both on terrestrial and otherwise. And again, that's especially an important consideration for diseases, pests, biosecurity issues, invasive pathogens, and so on. And so I guess what I'm going to sort of finish with really is just to argue that what we really need, and I think this is an applied science of coevolution. And in a sense, this seems to me this sort of nexus between ecology, evolution, environmental issues, and so on is exactly the right place for NCEAS and NCEAS-like activities to sit. So I think I'll just leave it there. Thank you.
My name's John Drake, and I guess my qualification, if there is one, for being on this panel, is that for me, NCEAS was the place that allowed me to discover the ecology of infectious diseases. So when I came here as a postdoc in 2004, I was interested in population dynamics. I figured that out already, and I wanted to find an important area of application that was full of all kinds of problems that might provide a lifetime of puzzles to figure out as a scientist. And infectious diseases is it. Before I was born, the Surgeon General reported to the U.S. Congress that the book was closed on infectious diseases. We know from the conversation that we've had already today that these reports of the demise of infectious diseases were indeed greatly exaggerated. And in fact, mortality due to infectious diseases continues to increase throughout the world, including in developed nations. I think that this mistake was made, and not just by the Surgeon General, by failing to recognize that the emergence and propagation of infectious diseases is actually an ecological and evolutionary problem. The majority of infectious diseases are organisms, and they change. And disease, pathogens, and their transmission is an ecological problem because it involves the interaction of species. Well, as it turns out, the world is also changing. The environment in which these interactions play themselves out is changing. And so that means that we have new and unforeseen problems that crop up because of this interaction of ecology and evolution. Largely due to the influence of NCEAS and ecologists who have taken an interest in infectious diseases, I think our picture of this as scientists and as a society has changed. So we've opened that book back up, and I think many institutions throughout the country and the world that are tasked with solving these problems are increasingly turning to ecologists for help doing that. So I guess I want to take a few minutes to reflect then on how ecology can do this going forward. Looking back, how is it that NCEAS was able to contribute so much? And what can we learn from that, anticipate about the problems that we have to solve in the future, and maybe articulate those problems so that we have collectively a clearer research direction. So what is the problem that we want to solve? Well, will the future world be a sicker world? Will the future warmer, environmentally fragmented, with lots of land converted to unfamiliar and novel uses, less diverse, wealthier, more densely populated and interconnected world? Will that world be a sicker world? Yes or no? And I'm part of the camp that says we don't know the answer to that question yet. Undoubtedly, that question is one that will require synthesis, and that's what NCEAS does really well, is synthesis. And not just any kind of synthesis, not just reviewing the literature and coming to some broad general consensus conclusion, but actually taking data and quantitative information and ideas and making them rigorous and precise and putting all those things together in a way that it can generate some, what I think people are nowadays calling actionable science. Science that speaks to the specific question that one or more many societies throughout the world would like together to answer. How does NCEAS do this, or how has NCEAS affected ecology so that ecologists do this? I think one of the main ways in which NCEAS has done this is by the creation and dissemination of new technical tools for synthesis. So these were all actually mentioned earlier today, so I don't have to bring them up, but let me just list what I think some of these are. One is formal meta-analysis. Meta-analysis is a way of taking lots of disagreeing analyses and finding the common thread that pulls them all together. And although meta-analysis existed prior to NCEAS, it was not widely used within ecology, and I believe now it is widely used. I think it's widely taught to graduate students. I think that lots of research teams and individual investigators are taking these tools and using them to great ends, and I think that that's borne out in the kind of review papers that you see coming out of ecological research. Another has to do with the combination of information from heterogeneous data sets. We have formal computational tools, some of which we call eco-informatics for doing this. And even in its sort of more informal manifestation, what working groups do is they bring together different pieces of information and they figure out how to sew them all up together. I think that's a much more common mode of doing research now as a direct result of NCEAS activities. And I think that's going to increasingly be important to solving this question, will the future world be a sicker world? The third is the use of models in order to precisely and specifically encapsulate an idea and bring those ideas to bear on, or bring data to bear on those ideas, to bring data and ideas together. Among other things, that can be done with sophisticated tools for model fitting. Things like MCMC, sequential Monte Carlo and iterative filtering, approximate Bayesian computation, and all kinds of other new cool gadgets for fitting models that were unheard of 10, 15 years ago. Model fitting has been around for a long time, but it was often a specialist enterprise, and I think now it's becoming something more in the mainstream. So with my last minute or so, I guess what I'd like to do is list a couple of things that I think might be particular technical problems for synthesis in the future. So for those of you that are involved in the future synthesis centers and in NCEAS 2.0, perhaps you might take some of these ideas and see whether or not there's anything that you can contribute or to advance our synthesis and answer this question, will the future world be a sicker one? The first is that I think we still need more principled means of combining qualitative and quantitative information. So we use meta-analysis and we use model fitting in order to bring different kinds of quantitative information to bear on a question. But often we know a great deal more than we're letting on when we set up our model and begin analysis. How can we do that in a rigorous, transparent, and repeatable way? I think that's a big problem for synthesis, and one that has some technical solutions, but they've not been found. The other big question is how do we extrapolate to future conditions? Will the future world be a sicker world? Presupposes what I think all of you probably agree, that there are important nonlinearities in this system. Whether you call them tipping points, phase transitions, bifurcations, or regime shifts, I think depends on your intellectual training and pedigree. But they're all the same thing. And the problem is the information we have is about the recent past, which has been more or less constant, and we think that's not going to remain. We think that it's going to change, and how to extrapolate to that future is a major problem for research. One of the things that we know as ecologists is that we can do that to some extent when we've dug into a system enough to understand its inner parts, the mechanisms that make it work. The question is, how can we do this in a substantially larger way, in a way that crosses the different scales between hosts, within hosts, and across landscapes, and across ecosystems even, in order to understand better the future of diseases in a continuously changing world.
Thanks a lot. And before I open it up for questions, I have one final thought and then a quote from Andy that I'd like to pass on as sentiment, basically. The thought goes back to something that Lou Gross said earlier. You've got synthesis, bringing together lots of data from different arenas to look at major patterns in ecological systems. You have analysis, things like fitting models to data, but also NCEAS has played a vital role in developing deeper fundamental theory. And I certainly, when I think back through the working groups in this area in particular, there are genuinely important novel contributions that were made to the theory of infectious disease ecology that came out of these NCEAS working groups that would have taken longer to have happened otherwise, I think. And I think that's an important, enduring contribution that NCEAS has made and we hope will make. The sentiment from Andy that I wanted to pass on, this was sort of goes beyond disease ecology. He says, quote, "I think the most important need for NCEAS is to continue to provide for the next generation of ecologists and other disciplines this beautiful independent place to meet, chat, work together, and foster future collaborations. And we need wonderful people like Jim Richmond and Sandy Andelman to run the place that is specifically for ecologists to synthesize, analyze, and create new theory." I think that's exactly on target. So with that, I will we'll start entertaining questions and discussions from the audience. Warren.
I'm Warren Porter from the University of Wisconsin-Madison. I have several comments here. I think that the comments about GMOs in relationship to infectious disease are not quite connected in the way that I understand it now. I know I'm known for a lot of energetics and heat mass transfer, but I've also been working in low-level environmental contaminants for the last 20-some years. And one of the things that we've discovered is that extremely low levels of chemicals can suppress your immune system, change your hormone levels, change your behavior and your learning abilities, and even your sexual orientation, and change developmental processes. The GMOs that we are seeing being introduced that are resistant to pesticides guarantee higher levels of those pesticides, which we know are suppressing immune function. If we're suppressing immune function, we are definitely going to be having more disease, I can guarantee you. We also know now, based on some new research by my wife and myself that we haven't quite published yet, that these chemicals that are associated with these GMOs are capable altering fundamental biochemical pathways, which result in all kinds of chronic, long-term, low-level disease implications. And if you look at the list of diseases associated with low-level immune suppression, that list is almost exactly coincident with everything the CDC is seeing going through the roof in terms of diseases in our population now. So that's point one. I also want to make a comment about model statistics analysis, especially in the context of very complex systems. When you're looking at a complex system like the biochemistry of the body, which has all kinds of nonlinear feedbacks, you cannot use your standard ANOVAs and other kinds of typical statistical analyses, because when you have more than about 10 variables, you can get any result that you want. And it's been a major problem in terms of individual variation, in terms of how we respond to disease. And we do respond differently individually. This has confounded a lot of the analyses of proteomics and the other kinds of omics that we're typically familiar with. There are technical ways that you can evaluate the impact of multi-variable systems that are highly interconnected, but you certainly can't do it with your standard statistical analyses. Thank you.
Yeah, Warren, I think I at least agree with what you said, and I'll hear what the other panelists say, but let me extract something more general from what you've done. When I think of what the current cutting edge in population biology is that when you're talking about populations, it's no longer sufficient to talk about numbers, density dependence, and so forth. That's necessary but not sufficient. What you need is to talk about individual quality, individual states. They can vary through time, they can be heterogeneous within populations. So in the case of disease, what you're saying is that exposure to certain toxicants can influence, very low levels of toxicants can influence immune responses, and therefore that'll influence a variety of dynamical parameters in a infectious disease population model. In like manner, varying the resource state of individuals, how well fed they are, can, in a variety of counterintuitive ways, influence disease dynamics by varying the internal states of organisms, either weakening their immune response or by altering the resources that the pathogens themselves are using as they grow and proliferate in the host organism. I think that's a very important area, and that brings out the importance of a clear understanding of what's going on at the level of the individual organism. Classical physiological ecology, the sort that you've done, and functional biology in general, is absolutely essential for population, community, and ecosystem ecology in dealing with these kinds of applications. Any other thoughts from panelists?
Yeah, go ahead.
It was almost simultaneously. So I just a quick comment. So I don't disagree with anything you said about GMOs. I mentioned GMOs for a couple of reasons, I guess. One, just as an example of the many kinds of things that are going to be happening on increasingly global scales because of some of these drivers like food security and other sorts of issues. And absolutely, I agree that a lot of these sorts of factors like GMOs, for example, are going to have all sorts of cascading direct and indirect effects. And some of these are going to be planned effects, and some of those are going to be unplanned effects. And the kinds of things that you're talking about are probably fall in that latter category of things that we didn't anticipate necessarily before we started deploying these things. And so I think they're absolutely critical for us to start uncovering those and understanding the consequences both in populations and communities and so on. And the other reason I guess I mentioned GMOs is that they're at least within GM organisms, particularly for things like herbicide-resistant BT cotton or crops like that, they at least represent examples where there has been some deliberate attempt to try to bring evolutionary principles into thinking about some aspects of managing those spatially and temporally, for example, in terms of the evolution of insecticide resistance in BT cotton, so that they're actually trying to manage that deliberately. And I just think it's an aspect of management of agroecological systems that we need to think about more broadly. So, but I don't disagree with anything you said.
Yeah, Warren, the thing that it brings to mind is that we've been in these climate-stressed ecosystems we've been working in. A lot of the root of infection is through immune compromise, and so what it means is not just the focus on the immune system, but the kinds of pathogens that are affecting the hosts suddenly you're dealing with these really nasty opportunists that are typically present and that are changing their behavior as the host's immune changes. And so that's an area where I'm not sure we really have developed very much theory. I mean, it's kind of a bit of a black box. So,
Jim.
Jim Brown. So I'd like to follow up on this very nice statement that John Drake gave and ask really the whole panel here. You asked the question whether the future world was going to be a sicker world. And my question would be, are we talking, are the concerns both for a chronically sicker world or an acutely sicker world? And what I haven't heard anything from this panel about is the possibility that this huge bubble of human population that we've created is going to be decimated by something like a global pandemic. Well, I know that's one of the most difficult things to predict, but it seems to me like it is a real challenge for our time, and I'd just be interested in hearing some comments from the panel about that.
Sure.
Well, I want to really thank you for that question. Well, so let's just take one pandemic, which we're concerned about, say flu. And where will ecology enter into this? Now, of course, the holy grail would be to be able to say that we're going to have a pandemic that's going to emerge on December 3rd, and be prepared. But that's not going to. So what can we do? We can try to figure out, based on ecological and evolutionary knowledge, what are the sources of potential pandemic outbreaks? How's flu being maintained? We actually don't know. So, for instance, if you take H3N2 as the model for the important flu that's coming out seasonally, you can use phylogenetic tools to be able to look at where the circulation strains are coming from, and they appear to be coming from Central and Southeast Asia. The question is, how does it persist in Central and Southeast Asia? And it looks like it actually may be persisting locally, since it seeds out seasonally. It may be persisting locally through metapopulation dynamics. Well, we don't really understand the metapopulation dynamics of infectious diseases. We usually look at single acute epidemics, and we can deal with them pretty well. But the metapopulation approach to understanding persistence is an important one. Now, it turns out that another pandemic that may be of concern is plague. So, and how does plague persist? Well, it turns out that plague may persist as a metapopulation. But the mechanisms of persistence of these two pathogens are very, very different. So we are starting to build a general theory of how infectious diseases may, in fact, play out as metapopulations with different kinds of persistence. Once we lay out those dynamics, then we can try to figure out what the appropriate intervention strategies are. So that's one, is the identification through an understanding of the dynamics of what the appropriate intervention strategy is. And the second is where the ecological dynamics really contribute, is in the development of targeted surveillance strategies. So you may not ever be able to predict that a pandemic is going to occur on December 3rd, but you can help guide the development of a surveillance structure that in fact maximizes the probability of early detection of a pandemic and therefore preventing it. Now I have to say that surveillance is a public health strategy. We can go on long with it, but the surveillance was identified. The Institute of Medicine did an important document called Microbial Threats in the United States. And they identified surveillance as the #1 thing we need to have worldwide in order to prevent pandemics. The strategy for surveillance in public health is entirely passive. You just sort of respond to what the information you're getting. Right now, there are efforts to develop more ecologically based, dynamics-based, infectious disease dynamics-based structuring of surveillance systems so as to maximize that. That's a whole new approach to understanding infectious disease. Homeland Security is interested in that strategy. The Department of Defense is supporting that. So I think that, well, to get to your question about will we ever predict a big pandemic, we'll be able to predict what the appropriate intervention strategies are, hopefully, and be able to build structures for early detection based on ecological dynamics.
Jim, several of us talked about how this is one area where you really have to fuse ecology and evolutionary biology. And one reason your question is particularly difficult is that it's not as if the properties of the pathogen are fixed and you're trying to figure out how it plays out. If that were the case, it would still be difficult. But they're moving targets. They're changing in response to the changes we impose in the environment. And so we need to know, Mike Hochberg, who's in the audience side, just submitted a paper with some people in France on, when can you make evolutionary predictions about the emergence of diseases when there's a constant trickle of spillover from a so-called reservoir host into other host species, where it seems very trivial at first. There's disease, but it's not really giving you the right kind of change of transmission, but evolution takes place so that, boom, you can be hammered very quickly by an outbreak. So this is one area where I think going back and forth between ecology, evolution, and genetics is absolutely essential. Other questions? Let's see. This gentleman's had his hand up for a while, I guess. Okay. And then Mary, who will be next? Okay.
I want to bring the, I'm David Bowman, I just want to bring the panel back to thinking about NCEAS, because you're certainly divergent from mainstream ecology. And that leads me to wonder whether NCEAS...
Excuse me, what did you say?
Well, it is, in all honesty. But that leads me to wonder with NCEAS too, is that maybe the word ecology is handicapped, and that what you're really talking about is a new kind of analysis and synthesis, which is necessarily evolutionary and ecological and holistic. And that would immediately broaden the constituency and the relevance and the reach and the impact, and let go of the idea that this is about bugs and birds and quadrates.
I think ecology is a large enough challenge to try to integrate in this way, but I think we have to, over the next 10 years or 20 years, I think there's going to have to be more synthesis across those major disciplines. I totally agree with that. I think that should be part of what NCEAS 2 is trying to do, one way or another.
I have to tell you a story. So I went to Emory 15 years ago, and Gene Odom was at the Institute. He was still alive at the Institute of Ecology. I was over there giving a talk, and he came up to me after the talk. I knew him fairly well, and he said, well, Les, Emory's never really been very big on ecology. They've really been very big on medical sciences. And he said, my daddy was the dean of the College of Arts and Sciences at Emory, and I'm really glad that they got an ecologist there now. And I turned to him and I said, well, Gene, I hate to tell you this, but my major collaborations are with the medical school and with the CDC. And he looked at me and draped his arm over my shoulder and said, well, if you can't beat them, join them. There's a real lesson in that, though, in joining them, which is a very good thing. In joining them, you still don't want to give up your identity, your intellectual identity, and your disciplinary identity. It's what we bring to that discourse. So it's not necessarily that we want to change our identity, because we have very specific knowledge and very specific ways of approaching and understanding the world, which is based on the development of a strong science. And it's the recognition that that science has a great deal to contribute, as well as its own scientific foundations can contribute to many, many important problems. But I would never suggest to give up that identity.
Mary.
So I wanted to return to the discussion of the apocalypse that the last three questions, and I've been thinking about this since your remarks on the lack of understanding of genetic linkages to disease dynamics. If the Dutch results hold up, and I'm sure you have much more up-to-date information at deeper levels than I, that we might be 3 or so mutations away from human transmissible 1918 type flu. If that's still true, then it seems that's kind of a game changer dynamic, linking the possibility of very rapid change that would have huge and abrupt population consequences for humans. I guess the other point on this is, that is NCEAS related more generally, is not that you want every scientist to immediately turn around and follow a particular lead or a particular bandwagon, but how does mega collaboration maintain its nimble ability to track really new information and even to discover new information and respond? Or is that something that should be at the level of individual laboratories? But I'm interested in just returning to Jim Brown's question and hearing what maybe the latest is on that Dutch result. And whether it could, it seems as if it's true, it's a very simple, amazing connection between genetics and disease dynamics.
No? I'm not aware of that.
See, that's the problem with meta.
Yeah. Not quite simple.
They're drawing a blanket.
Just, Mary, you were talking about these sort of mega collaborations, which I think is inevitable for a lot of these problems. But I think part of that has to be, I think the model we had was where you had a succession of interlocking, partly overlapping working groups with some postdocs and some saboteurs and so forth. It was very that come out wrong. Was quite effective, actually. And I think that has to be maintained. The problem with big often comes with bureaucratic rigidities. And you don't want that. You want to foster individual creativity, ultimately, right? So I think NCEAS has been very effective at that. Luke.
Okay. All right, I'll wait. Thank you. Lou Gross from Nimbus. So I notice a theme that underlies many of your conversations, as well as Warren's back here, and that is that it really is time for ecology to assert its importance at the same level that omics has asserted its importance to human health issues. You've talked about the landscape of the pathogen within the individual, and one possible route for a future NCEAS is the focus on the individual as the center of interest for many of our federal agencies, not just NIH and the human body, but pretty much all of agriculture, many of the DoD issues that deal with deployed military personnel. There are many, many aspects that could really benefit from an ecological perspective of the individual. And I mean beyond the microbiome issues. So that's my view of one way that NCEAS could move forward by having a focus that would be of interest to a variety of agencies.
Let's see, Warren, that's sort of your bailiwick.
Thank you. I just wanted to comment on the dynamics of flu. The reason that these flu viruses keep popping up in different areas in Southeast Asia is because of a social situation in terms of the farmers that live in that area. They typically will raise both chickens and pigs very close to each other in proximity. Now it turns out that bird flu viruses, receptors for those don't happen in us, in humans. But pigs have both types of receptors, one for both birds and the other one for mammals, for humans. And so the transmission of new virus that might have been developed because birds are migrating back and forth and carrying stuff and promoting evolutionary change gets to us because of that chicken-pig-human connection. It gets from the bird to the pig and then from the pig to the human, and that's how these new things get started. I just thought you might like to know that there's a social part of this whole equation in terms of these flu dynamics.
Yeah, that's absolutely true, and there's a great deal of interest in looking at social dynamics in disease epidemiology and the spread of infectious diseases. So just as a interesting side where social dynamics become clear, for instance, is there was a great deal of concern about what happens when you have an outbreak. We haven't taken into account, for instance, human fear and how that induces movement. So what happens when people get scared? How do they move across the landscape? And what does that do for the spatial dynamics of the disease? So fear as an element of an epidemic model is something that needs to be figured out how you incorporate that. But the ecological, the interesting intellectual ecological problem, there are the specifics of every disease. But so for instance, where the ecological theory comes in, for instance, is in plague, which is a vector-borne disease. Turns out that the bacillus associated with plague can persist for a very long period of time on fleas, which remain locally present, even though the entire population of its host has died out. The fleas are just sitting there with the bacteria, and then they get recolonized by hosts, which is a decidedly different kind of, it's a very lethal disease, which is a very different kind of mechanism than, say, rabies, where all the hosts, raccoon hosts, for instance, die, and the metapopulation, locally the population dies out, and the persistence mechanism is through induced movement of infected individuals, and they have a longer duration of infectiousness. So it's trying to figure out how the fundamental parameters which guide all epidemic models lead to different kinds of stable dynamics along larger landscapes. And we really don't know that in a way that manifests itself in a variety of diseases, and we know that that's starting to be important. That, in order to take that approach, just to counterbalance Lou's point, in order to understand that approach and take that approach, you're not going to be able to do that at an individual level. There are questions that are going to be very specific to at the individual level, but we definitely need to have a population approach as well.
Absolutely. Let's see, there's a question all the way in the back. I can't see who it is. Just see it.
Cynthia Stebbins, and I'm a guest here, so I thank you all for that. It's a privilege. I want to speak to what the gentleman in the red and white checked shirt had to say and add to it a bit. There are all sorts of instances where pigs have been a huge factor, most especially now in Malaysia and Bangladesh, where Nipah virus is particularly virulent. And that came about largely because the pig farmers decided they wanted to have factory farms and move further into areas that hadn't been used for agriculture, especially pig farms before. And it wasn't chickens, it was bats, flying foxes, that would come into the odd fruit trees that were left to provide shade to these pig pens, and they eat a little bit of fruit and then drop some, and the pigs, of course, would eat it and develop what was called a 1 mile cough, which is how that one spread. And Nipah virus, Hendra virus, tends to spread the same way. So those are all factors, but I also wanted to mention the fact that many years ago, the head of the organization where Andy Dobson and I were on the board together called a press conference, and she announced, her name is Mary Pearl, and Mary announced to the gathered press that we were about to have a problem with zoonotic diseases that no one had really recognized before. And no one paid very much attention. There wasn't much coverage in papers or anything. But 5 months later, crows began to fall out of the sky. And we all recognized that Mary was right on the money. So there are organizations that are dealing with this now. And hopefully you can all get together, because it seems silly for everyone to be, it's just redundant if one isn't compiling that information and working together.
Yeah. this is sort of a lateral comment rather than anything directly related. I agree with what you said. And that is, the last few questions have all focused on diseases of direct impact on humans and our commensals, which is, of course, where we start. That's where the money is. That's NIH. That's medicine. But the other contribution NCEAS has made that I think is still very important is that just understanding the role of pathogens and parasites in natural ecosystems. And there's a much broader diffuse problem out there. You know, I'm shocked every time I go into the woods with a good naturalist. I hear about some new tree that's being hammered by some pathogen. You know, the Persea bays of Florida are all dying out because of some pathogen that's been introduced. You know, dogwoods have been hammered. You know, we were just talking about something that was introduced to Australia that has a huge host range, and all these things are going to be aggravated by climate change. So I think there's an enormous dimension to infectious disease ecology that goes well beyond human health, but has to do with the fundamental integrity of natural ecosystems, coral reefs, forests, and so forth. And I think that that's something that, assuming NCEAS 2.0 still has a substantial infectious disease component to it, I think that would be a really valuable contribution to make to further delve into that. Are there any other questions? Well, if not, 15-minute break. 15-minute break. Great.